Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs for the year 2022 from Genetics section. If you are new to my channel, please do like, share and subscribe. Coming to February 2022 question paper, shift 1. So first question, a fly with apricot colored eye was crossed with a sepia-eyed fly of opposite sex. In F1, all flies were wild type. The genes responsible for the two phenotypes were. So here F1 resulted in wild type organism when both the parents are having, that means both the parents are having mutated eye color. Now this is a case of complementary gene interaction. So they are non-allelic. So the genes responsible for the two phenotypes are non-allelic. So the answer here is option 2, non-allelic. Next question. DNA was isolated from a strain of bacterium with genotype A plus B plus C plus D plus E plus and transformed into a bacterial strain A minus B minus C minus D minus E minus. The transformants were tested for the presence of the donated genes. The co-transformed genes were found as follows: A plus and B plus, C plus and E plus, D plus and C plus, B plus and D plus. What is the order of the genes on the bacterial chromosome? So here if you see, A and B go together. So A plus, B plus. Then along with B, D is going. So B plus, D plus. So A plus, B plus, D plus. D is going along with C. So A plus, B plus, D plus, C plus. Then C is going along with E. So A, B, C, D, E is the gene order. Answer is option 4. Then next question. Human polydactyly traits having extra fingers or toes are caused by a dominant allele. In a screening, it was found that out of 42 individuals having an allele for polydactyly, only 38 of them were polydactylous. Which of the following is the correct interpretation of the observation? So this is a case of penetrance. So if you see the penetrance of polydactyly, you can see that out of 42 individuals, 38 of them were polydactylous. So 38 by 42 into 100, you will get around 90%. So the penetrance of polydactyly is estimated to be 90%. Answer is option 1. Expressivity of polydactyly is 90%. Expressivity is not a quantified value. It cannot be quantified. Then this is an example of variable expressivity. That is also not correct. The polydactyly trait is showing complete penetrance. If it was showing complete penetrance, then 42 out of 42 individuals would have been polydactylous. But here only 38 of them are polydactylous, that is 90% of them are having polydactyly. So it is not a case of complete penetrance. So the answer here is option 1. Then next one. A group of transposable elements described as retroelements encompass. So out of these, P elements in Drosophila, lines and but not signs in human. Lines and signs are also retroelements. So if you see the third option, that is copia elements in Drosophila, lines as well as signs in human. So answer here is option 3. Then next one. 22 transduction was used to map the FADL gene. The result of the two-factor crosses between FADL and two linked markers PURF and AROC are shown below. So this is the donor, recipient, this is the markers, and these are the numbers obtained. So from the donor, if you see, first we have to see for the transduction frequency. So if you see the donor, what are the genes? FADL and PURF. So here you can see that PURF and FADL, the numbers obtained are 200. So they are showing 20% frequency of transduction. How did you get 20%? That is 200 plus 800 is 1000. So out of 1000, 200 of them are transformants. So if you convert it into percentage, you will get it as 20%. Now the next condition, FADL plus AROC plus. If you see AROC plus and FADL, it is 400. So here it is 40% frequency. Now here if you see AROC plus PURF. Now if you see it is 500 over here. So 500 means 50% transduction frequency. So the least one which is having less percentage of frequency is PURF plus and FADL. So that means they are far apart. So PURF, FADL far apart and they are 20 centimorgans apart. Now if you see FADL and AROC plus, you see it is 400 and AROC plus and PURF it is 500. So the highest is for AROC plus PURF, that is 500. So AROC plus and PURF will be nearby. So because they have the highest percentage of frequency of transformation, so they will be nearby. So it will be 50%, and the next will be AROC plus and FADL, that will be 40%. So the ones which are at the ends are PURF and FADL, and AROC will be in the middle. So such kind of graph you can see. Yes, that is seen in option number 2 of these given options. So FADL and PURF are at the ends and AROC in the middle. So FADL and PURF least frequency, so they are far apart, that is 20%, then comes 50% and then comes 40%. So the answer here is option 2. Then next one. The pedigree given below represents the genotype of 4 different loci for children in generation 3. Which of the given genotypes is likely to represent the genotype of individual 2-1? So this individual they have asked, that is the mother or the female. They have asked what is the genotype of the individual. Now if you see this, you can see that they have given 2 chromosomes for females and only 1 chromosome each for male. So that itself shows that it represents an X-linked inheritance. And we know that it is X chromosome genotype. Okay, X chromosome genotype that they have, that is given, and X chromosome in males it is obtained from only the mother, and here it is 2-1. So the mother is giving these 2 chromosomes, and we know that recombination of between the X chromosome is also there. That you have to keep in mind. Recombination between X chromosome is also there. Now if you see these 2 genotypes, this one A2 B1 C2 D1, and in 3-2 also A2 B1 C2 D1, they both are from the father. Why? Because there won't be any recombination. Father is having 1 X chromosome and 1 Y chromosome, so recombination chances are very, very less. Will not take place also. So these 2 genes, so these 2 genotypes have been obtained by 1 and 2, 3-1 and 3-2 from the father, that is 2-2. So that represents the genotype of the father. Now we have to find out the genotype of the mother. Now these are X chromosomes which are obtained from the mother, that is 2-1. Now if you see these 3 and 4, you can see that the female is having A1 and A2, B1 and B2, C2, C1, or it can be even C1 and C2. If recombination has taken place, then C2, C1. If not, it is C1 and C2 also. So there is A1, A2, B1, B2, C2 C1 or C1 C2 and D1 D1. Now this D1, it can be obtained either through recombination also. It can be either D1 and D2 also. Now if you see this one 3 2, you can see that there is a D2 over here. So chances are there that this D2 and D1 might have undergone recombination of their X chromosome, and one X chromosome went to the male child and one X chromosome went to the female child. So the genotype of the parent will be, of the mother, that is 2 1 will be A1 A2 B1 B2 C1 C2 D1 D2. So that can be seen in option 3. A1 B1 C1 D1 A2 C2 D2. So the answer here is option 3. Then next one. A Drosophila mutant line with vestigial wings is isolated in a laboratory. The vestigial wing phenotype was observed to be recessive and mapped to gene X. This was mapped to a gene called as X. It is not X chromosome, it is just gene X. As we say gene A, gene B, gene C, just like that gene X. 3 other laboratories also isolated vestigial mutants called line B, C and D. In order to test if the mutation in lines B to D also mapped to gene X, the following crosses were made and phenotype of F1 progeny observed. Based on the above, identify the lines which is most likely not to have a mutation in gene X. So here what they have done is a Drosophila mutant with vestigial wings, and vestigial wing phenotype was observed to be recessive, and this phenotype was mapped to gene X. Now 3 other laboratories, they also isolated vestigial mutants and they named them as line B, line C and line D. Now they too want to see whether this particular B to D they mapped to gene X, so they made the following crosses. A into B gave rise to vestigial wings, A into C gave rise to vestigial wings, AD cross gave rise to normal wings, BC gave rise to vestigial wings, and BD gave rise to normal wings, and CD gave rise to normal wings. Now if you see the data that they have given, you can see that whenever there is a cross with D, whenever there is a cross with D over here and over here, that is when A is crossed with D, B is crossed with D and C is crossed with D, you get normal phenotype or normal wing morphology. That means when D is crossed with a mutant, you get only a normal phenotype. So D does not have any mutation in gene X. So the answer here is option D. C, line D only. Now coming to the next question. Wheat plants can have kernels of white color or in shades of red. That is light red, red, dark red, very dark red. That is purple. A researcher made the following crosses. So wheat plants with white kernels were crossed with wheat plants with purple or very dark red kernels. So here you can see the F1 phenotype was all plants with red kernels, and F2 phenotype gave it in the ratio 1:4:6:4:1. That is plants with purple kernels, plants with dark red kernels, plants with red kernels, plants with light red kernels, and plants with white kernels. So if you see the offsprings of the F2 generation, we come to know that it is a case of quantitative inheritance and caused by more than one gene, because the phenotypic effect of all the genes together is responsible for the formation of the different color pattern in wheat kernels. So here the answer is going to be the two genes, both coding for the color of kernel and each gene having two alleles, one that produced red pigment and the other that produced no pigment. That is, B is going to be the answer. If you see the statement A, it is a dihybrid cross. Yes, of course it is a dihybrid cross where white color is coded by A gene and the purple color is coded by B gene. Here both the genes, that is A and B, they jointly give the color. So it is not that white color is coded by A gene and purple color is coded by B gene. So that is not the correct answer. 4 genes, one coding for no pigment. Only 2 genes are involved, not 4 genes. The trait is influenced by environment leading to the observed variation in the kernel color. That is also not correct. Now I have done the dihybrid cross and cross for the Punnett square for this dihybrid cross, and it is given in the next slide. I will show it. So this is the individual, that is AABB, that is purple, and small a, small b is white, and these are the F1 offsprings, and F1, if you cross it, you will get F2 individuals where capital A, capital B is purple color. That is 1 out of 16 will be purple color and 1 out of 16 will be white color, and these are the dark red individuals, these 4 are the light red individuals, and these, that is red, these are the red colored wheat kernels. So this is how the cross will be. So this is a case of quantitative inheritance and involves 2 genes. So coming to the next session, that is shift 2, February 2022. First question. A newborn baby got mixed up with other babies in a hospital. If the mother is O blood group and Rh positive and father is AB blood group and is Rh negative, which one of the following can be their baby? So, if the mother is O blood group and is Rh positive, so this is the blood group of the mother, that is O blood group and Rh positive, and this is that of the father, AB and Rh negative. So this will be the gamete produced by the mother, and these are the 2 types of gamete produced by the father. The child will be either AO or BO and Rh positive. So A positive or B positive. So if the mother, which one of the following can be their baby? So AB, it cannot, first option is AB and Rh positive. So it cannot be AB. Then if you see O, it cannot be O. If you see A, yes, it can be A, Rh positive. So answer here is option A, option C, that is A and Rh positive. It cannot be B and Rh negative. The next one. 2 genes A and B are located at a distance of 10 centimorgan. Individuals of the genotype AABB are SIB-mated. SIB-mated means they are selfed. The 2 genes are linked in trans. So if you have capital A and capital B and small a and capital B, that is in case of trans linkage, that is like this, AB, AB. What percentage of the progeny is expected to have the genotype small a, small b, small b? So these are the genes, 2 genes A and B. They are located 10 centimorgan apart, and individuals, that is A, capital A, capital B, small b, they are selfed. So these are the individuals that are crossed, and you get these as the gametes, that is capital A, capital B and small b. So capital A, capital B and small a, small b. Now they will be present at 5% frequency. So small, they have asked for percentage of the progeny expected to have genotype small a, small b. So if you cross these two gametes, that is small a, small b and small b over here, you get 5 into 5%. That is, these two should come together. Only then you will get the genotype small a, small b. So 5% into 5%, that is 25%. So it will be 0.25. So the answer here is option A. Then next one. Which one of the following statements is true regarding heritability of a quantitative character? So they have asked here is broad sense heritability, and broad sense heritability is given by the formula genotypic variance divided by total variance. So variance in the genotype divided by total variance. So the answer here is heritably measures the proportion of the phenotypic variation that is the result of genotypic factors. So the answer here is option C. Then next one. Double haploids are plants derived from single immature pollen and doubled artificially to form diploids. A double haploid population was created from F1 progeny derived from a cross between two parents P1, P2, this is the F1 progeny, one resistant and the other sensitive to white rust. The parents F1 and DH population were profiled with a set of codominant markers which is represented below. The following table summarizes the proposed percentage of the four different types 1 to 4 of double haploid progeny. Assuming that the DNA marker is unlinked to the gene governing the trait and linked at a distance of 10 centimorgan, which one of the following options correctly represents the expected ratio for unlinked and linked respectively? So if you see the case of unlinked, you can see that there is equal probability of occurrence. So here if you see A, it is 25, 25, 25 in the case of unlinked, and 37.5, 37.5, 12.5, 12.5. You cannot get 12.5, 12.5. It should be all of the same percentage. So A is correct for unlinked condition. Now if you see the case of linked, so A and B are linked. So these are the gametes that you will obtain from this cross. There will be two recombinants and they will be 5% each. The rest will be parental phenotype, this and this, they will be 45% each. So this is the case you get with linked at a distance of 10 centimorgan. So the first option, that is A. If you see for linked, it is 45, 5, 5 and 45. So the answer here is option 2, that is A1. Then next one. A Drosophila male carrying an X-linked temperature-sensitive recessive mutation that is lethal at 29 degrees Celsius but viable at 18 degrees Celsius is mated to a normal female. Then B, a female containing attached X chromosome. If the eggs laid in both the cases are reared at 29 degrees Celsius, what will be male-female ratio in the given progeny? So a Drosophila male, it is carrying an X-linked temperature-sensitive recessive mutation, and that mutation, it becomes lethal at 29 degrees Celsius and it remains viable at 18 degrees Celsius. That is, at 29 degrees Celsius, the offsprings will be killed, and if it is at 18 degrees Celsius, they will be viable. So this Drosophila, it is mated with a normal female. So XS, that is Drosophila sensitive to with the temperature sensitive mutation. XY is the male and XX, that is normal female. Now if you cross, you will get 1:1. Here the sensitive S mutation has gone to the female, and in female there are 2 X chromosome, and the normal X chromosome will suppress the mutant X chromosome, so this will survive. So A will be 1:1 ratio. Now coming to B. B is a female containing attached X chromosome. So the attached X chromosome I have represented like this, and this is the male Drosophila. Now the attached X chromosome, as such it will go to the female, and this XS will be obtained from the male Drosophila in the female, and female will be having 3X, and only Y will be there in the case of the second offspring. It will not survive. So the answer here will be option 2, that is A 1:1, and B only females survive. So the answer here is option 2. Now next one. In 1990, Bhattacharya et al. Identified that the wrinkled seed character of P, as described by Mendel, is caused by a transposon-like insertion in a gene encoding starch branching enzyme encoded by the R allele. This leads to an RFLP pattern when genomic DNA of round and wrinkled seed is digested with ECO-R1 and probed with cDNA of the R gene product. The following is a representation of the hybridization pattern. Based on the above, which of the following statement is incorrect? So first option, lane 3 represents genomic DNA from plants with wrinkled seeds. So if lane 3 has wrinkled gene, then the genotype should be smaller, and you should get a single band and not 2 bands. So the first option itself is incorrect, and the answer is first option. Then next one. In recent decades, the use of genetic markers has allowed the rapid introgression and selection of desired breeding stocks in advanced generation. In this regard, the following statements are given. AFLP markers can discriminate between homozygote and heterozygote genotype. Now AFLP is a dominant marker and it cannot discriminate. So that option is not correct. Then microsatellites, example SSR, are capable of detecting higher level of polymorphism than RFLP. That is correct. Then next one. SNPs are more prevalent in the coding region of the genome. They can be found throughout the genome. So that is also not correct. Then SNP markers are most suitable markers for both foreground and background selection. That is also correct. So the answer here is B and D, and that is option 4. A cross is made between Drosophila stocks, each with an independent mutant allele resulting in white eye color. The mutant alleles named W1 and W2 are recessive, X-linked, and caused by deletion in the W plus allele. The wild type phenotype is red eye color. The F1 females are then crossed with wild type males. So this is the Drosophila mutant having the alleles XW1. One moment. The mutant allele, the F1 females, are then crossed with the wild type male. So this is the F1 female. Okay. XW1, XW2, and it is crossed with the wild type male, XW plus and Y. Now these are the gametes that are obtained. On crossing you will get XW plus W1, which is wild type female, then W2 and W plus, so again a wild type female, whereas the males will be mutants. So what did they observe? in the progeny, all females have red color. 1 out of 10,000 males was observed to have red eye color. I told you that after the cross, it is white eye color, that is mutations are found in the males. But here they observed 1 out of 10,000 males was observed to have red eye color, while the remaining had white eyes. Which one of the following could possibly explain the occurrence of red-eyed males in the progeny? So that is a case of recombination between the W1 and W2 alleles. That is why the males got red eye color. That is 1 out of 10,000 males got red eye color. So the answer here is option B. There is intragenic recombination between W1 and W2 alleles during meiosis of F1. Coming to the year September 22, shift 1. So coming to the first question. Given below is a pedigree indicating a pattern of inheritance. So which of the following option correctly describes the pattern of inheritance shown in the above pedigree? So if you see the pedigree, you can see that this generation is not having the disease, whereas these offsprings of that generation are having the disease. That means it is skipping the generation. So it should be autosomal recessive. But if it is X-linked recessive, then father should have been deceased. So the answer here is autosomal recessive. Then next one. In a conjugation experiment between bacterial HFR strain X and F- cell, lac gene enters the recipient in 4 minutes. So that is the first gene to enter. But the F- cell remain auxotrophic for leucine, tryptophan, URA, glycine, glutamic acid, phenylalanine. The mating is then allowed to proceed for 20 minutes and the lac plus X conjugates are selected. Of the lac plus cells, select the correct order of the genes as they enter from the choices given below. So this is the case of conjugation experiment in bacteria, and here the first I told you that the lac plus gene is the first to enter. So lac plus X conjugates are selected, so lac gene has to come first. So if you see the options, you can happily eliminate A and option 1 and option 3 because their phenylalanine and tryptophan are the first to enter and lac is last to enter. So that means those options are not correct. Then out of second and fourth option, which is the correct option? So after lac plus, the next one with higher frequency to enter was tryptophan. So after lac plus comes tryptophan. Then after that comes glycine. Then after that comes glutamic acid 65%. Then leucine at 35%. URA at 10% and phenylalanine at 0%. So your option is option D or option 4. Then next one. The following picture represents a gel profile of a pair of DNA markers observed in parents P1 and P2. They are F1 progeny and F2 progeny. 4 different profiles were observed in the case of F2. The number of F2 progeny showing a given profile is indicated in brackets. So this is the F2 progeny. The number of F2 progeny is given in the brackets. If you see it, you can see that it is approximately equal to 9:3:3:1 ratio. So out of these, which is the correct option? So if you see, read the options, you can see that the polymorphic DNA bands represent 2 independent genes. That is why they are in the ratio 9:3:3:1. So the answer here is option 2. Then next one. The additive nature of genetic map as suggested by Alfred Sturtevant and T.H. Morgan is possible if there is if there is complete interference in crossovers. So answer is option 2. Then next one. The following represents sequences of different alleles of a gene found in a family represented by mother, allele 1, allele 2. Father, allele 1, allele 2, and there are two sons, allele 1, allele 2, son 2, allele 1, allele 2. Further, a new mutation was observed in one of the alleles of the son, which is marked with a triangle. The following statements were made about the mutation. The mutation arose in the germ line of the father. These are the alleles for son 1 and son 2. Now if you see this allele, CAG, CAT, CAT, AGT, if you see that, you can see that it resembles mother allele 1. That is this allele, CAG, CAT, AGT, CATT, CGT. This allele represents mother allele 1. Similarly, here also if you compare this son allele 1, son 2 allele 1 with father and mother allele, you will see that it represents mother allele 2. That is this allele. Is similar to, is same as this allele. But if you see this one, where they have got a marked a mutation in the form of a triangle, if you see that one, we have to see from where that allele has come to son 1. So CAG, CTT, if you see that it is father allele 2, it is same as father allele 2. CAG, CATT, AG, TC, ATT, CGT. So it is same as father allele 2. So and son allele, son 2 allele 2, that also is father allele 2. So the mutation here has arose in the germline of the father. So option statement A is correct. The mutation arose in the son. No. The given DNA sequence are present on the X chromosome. So that is also not correct. There is a possibility to use RFLP tracking this variation. So that also is correct. So A and D are correct, and the option here is, and the answer here is 370. Option ID 370, that is A and D. Then next one. In Drosophila, balancer chromosomes are used to keep all the alleles on one chromosome together. A balancer contains multiple inversions so that when it recombines with the corresponding wild type chromosome, no viable crossover products are formed. Balancers also carry an allele for the dominant phenotype. Now a Drosophila male with sepia eye color is crossed to a female carrying a third chromosome balancer. The allele for sepia phenotype is located on chromosome 3 and is recessive to the wild type eye color. The dominant marker for TM6B is a tubby phenotype. Further, an individual homozygous for TM6B balancer does not survive. F1 progeny with the tubby phenotype is sigmated. The F2 progeny is expected to have. So here, a Drosophila male with sepia eye color is crossed to a female carrying a third balancer chromosome, and the dominant marker for this TM chromosome balancer is tubby phenotype. And further, an individual homozygous for this tubby phenotype does not survive. So F1 progeny with the tubby phenotype is self. This is the F1 progeny TT, that is tubby phenotype. That is the F1 progeny, it is self, and you get 3 different progenies, of which capital T will be dead, because they have mentioned already an individual homozygous for TM6B balancer does not survive. So that will be dead. If you see for sepia eye color, they have asked for sepia eye color only, not the wild type eye color. So sepia eye color, it is in the ratio of 1. So sepia and tubby flies in the ratio 1:2. So the answer here is option 3. Then next one. Given below is a pedigree indicating a pattern of inheritance. So this is the pattern of this is the pedigree that they have given, and they have given following statements are drawn from the above pedigree towards understanding the pattern of inheritance. And first of all, we will see what the pedigree is. So if you see the pedigree, you can see that whenever a male is affected, the female is a carrier. So naturally it has to be an X-linked recessive trait. And if the female is a carrier and male is not infected, only you get a carrier female. But okay, so this is an X-linked inheritance. The following statements are drawn from the above pedigree towards understanding the pattern of inheritance. An affected male does not appear to pass the trait to his sons. That is correct. An affected male appears to pass the allele to a daughter who is unaffected. Daughter is carrier, so she is unaffected, so that's also correct. All individuals have at least one affected parent. No, if you see the case here, these are not affected, but there is an affected son. The given trait appears to be a recessive one. I told you it is X-linked recessive, so naturally that is also correct. The given trait appears to be an autosomal recessive one. So that is also not correct. It is X-linked recessive one. So A, B, D are correct. So the answer is option 2. Then next one. 2 new chemical compounds X and Y are synthesized in the laboratory and tested for their potency as a mutagen. The nature of the mutation produced by these compounds is tested for reversal by other known mutagens, and the following results were obtained. Which statement best describes the nature of the 2 mutagens? So you have a new compound X and Y. It is reversed by 5-bromouracil, EMS, hydroxylamine, acridine orange. So compound X, it is reversed by 5-bromouracil and EMS. And 5-bromouracil and EMS, they are known to cause transition. So X is a compound that produces transition. So Y, it is reversed by acridine orange. And acridine orange is known to cause frame shift mutation or insertion deletion mutation. So Y is a compound that produces insertions or deletion or frame shift mutations. So the answer here is compound X produces single base substitution that generates CG to TA transition, and compound Y produces insertions or deletions. So answer is option 1. Then coming to shift 2. AFLP technique generates polymorphic DNA fragments that are generally scored as dominant markers. However, a pair of DNA fragments, say A and B, generated by AFP can be termed as codominant if on analysis of a large progeny of double haploids derived from an F1, it is observed that 50% of the progeny has fragment A and the remaining has fragment B. So the answer is option 2. Then next one. In which one of the following human disorders parents or grandparents are said to carry pre-mutations? So Down syndrome, it is trisomy of the 21st chromosome. So that is not the answer. If you see Klinefelter syndrome, that's also a chromosomal disease, chromosomal abnormality. And alkaptonuria, it is an autosomal recessive condition, and in autosomal recessive condition, unaffected parents can produce affected child. So what remains is Fragile X syndrome. Affected parents produce affected child, and it is an X-linked dominant trait. So the answer here is option 2. Then next one. If a gamete produced following non-disjunction of a chromosome at second meiotic division was fertilized by a normal gamete, what is the expected frequency of trisomic progeny? So the gametes, what are the type of gametes that you get if there is a non-disjunction of chromosome at second meiotic division? You get two gametes that are N, one gamete that is N plus 1 and one gamete that is N minus 1. Now, what is the expected frequency of trisomic progeny if it was fertilized by a normal gamete? So N plus 1 plus N will give you 2N plus 1, that is trisomy. So 1/4 of the individuals will be trisomic. So answer here is 190, option ID 781. Then next one. How many complementation groups do the following mutants M1 to M6 come under? So first what we have to do is, those that do not complement each other, place them in one group. Now M1 do not complement each other. That is why they have given a minus mark. So M1, then M1, M2 and M1, M5. So M1, M2, M5 in one group. Then next one, M2 we have already written. M5 also already taken. Then M3 and M4, that's another group. M4 we have already taken. M5 also already taken. M6 is the third group. So how many complementation groups you have? 1, 2 and 3. So the answer here is option 3. Then next one. Fertilization between two mating types P1 and P2 of Neurospora led to a diploid ascus cell, which gave rise to ascus containing 8 haploid ascospores. A set of DNA markers representing two linked loci was analyzed in P1, P2, and the octets labeled 0.1 to 8 arranged from the tip to the base of the ascus. The observed profile is represented below. Which one of the following is a correct conclusion of the above observation? So fertilization between two mating types, that is P1 and P2, you will get two ratios, that is first division segregation you will have 4:4 ratio. Second division segregation you will get two types of ratios, that is one is 2:2:2:2 and the other is 2:4:2. Now we will read the statements. Bands labeled A and C are allelic and segregation occurred during meiosis 2. So A and C, this is A, this is C, this is A band, this is C band. So it is in the 4:4 ratio, 1, 2, 3, 4, 1, 2, 3, 4. So it is 4:4 ratio and it is first division segregation ratio. So it should be meiosis 1 and not meiosis 2. So first option is not correct. Bands labeled B and D. These two bands are allelic and segregation occurred during meiosis 2. So B and D are allelic, that is okay. B 2 here, 2 here. D also 2 here, 2 here. So it is 2:2:2:2 ratio. And segregation occurred during meiosis 2. That's correct because the 2:2:2 ratio is a second division segregation ratio. So option 2 is correct. Now next one. Bands labeled A and D are allelic and segregation occurred during meiosis 2. Now A is here, D is here. So these are the A bands and these are the D bands. So A and D, they come together, so they are not allelic. So option 3 is not correct. Then last option, bands labeled C and D. These two are allelic and segregation occurred during meiosis 1. So if you see bands labeled C and D, C and D, that is also they are not allelic, so segregation does not occur during meiosis 1. So that option is also not correct. So the correct answer here is option 2. Then next one. During development, many gene products are provided by the female to the eggs which are needed for normal development of the zygote. Such genes are called as maternal effect genes. The following are set of crosses between parents carrying a recessive mutant allele M and the offsprings obtained. Which of the above crosses is or are indicative that the mutation is in a maternal effect gene? Now, whenever maternal effect is shown, genotype of the mother will decide the phenotype of the offspring or the progeny. So here what you have to see is, you have to see the genotype of the mother and then check with the phenotype of the offspring in the table. So you have to see for only these ones. These ones. This is the male parent and this is the female parent. So you have to see for the genotype of the female parent. So M plus, it is wild type, phenotype of offspring, all normal. Here also MN plus, all are normal. MM mutants, so all mutants. MM all mutants, all mutants. M plus, both normal and mutant. So you can see that along with the normal phenotype there are mutants also, showing that maternal effect genes got mutated in some of the progeny. So the answer here is option 2, cross 5 only. Then next one. The pedigree below is in reference to Engelmann syndrome, which is caused by mutation in the UBE3A gene on chromosome 15. The gene is also paternally imprinted. Individuals showing Engelmann syndrome have not been indicated in the given pedigree. Individual 1-1 does not have Engelmann syndrome. Individuals marked with the dots are carriers for UBE3A mutation. Which one of the following option list individual, all of whom are likely to show Engelmann syndrome? So if you see here, that is 2-2 and 2-3, these two. If you see here, this is a carrier mother and she has given her genes to son and her gene has also gone to the daughter, and from there the father is not a carrier. He is giving his imprinted gene to the next generation or to his offsprings. So from here the imprinted gene goes from the father to the son, imprinted gene goes from the father to the daughter. So they both will be deceased. So 3-1, 3-2, they both will be deceased. Similarly, the case over here also, here 4-2 will be deceased. But if you see here 5-2 and 3-5, if you see those two, this daughter will not be affected because she is getting the mutated gene from the father itself. So she will not be affected. So the answer here is 3-1, 3-2 and 4-2. 3-1, 3-2 and 4-2. That is seen in option 2. So the answer here is option 2. Then next one. 4 different HFR strains of E. coli were mated with F- recipients and the time of entry of various donor markers were found to be as below. HFR1, methionine 15 minutes, threonine 30 minutes. All these I have mentioned in the next slide. Based on the above observation, the following statements were made assuming met to be at 0 minute and threonine at 15 minutes. So histidine is located at 50. I will give you the answer, then I will give the explanation. Histidine is located at 59 minutes. That is correct. Then ASI is located at 74 minutes. That is also correct. Then cysteine is located at 11 minutes. That is also correct. MAL is located at 76 minutes. That is not correct. So this is the HFR strains. So methionine 15, threonine 30 minus. If you minus 30 from 15, you will get 15 minutes. So methionine and threonine are 15 minutes apart. Similarly, threonine and phenylalanine 12 minutes. MAL and phenylalanine 15 minutes. Then the HFR strain 2, that is bio and thymine, are 1 minute. Similarly, if you calculate for all these, you will get a graph or a map like this. Methionine, threonine, phenylalanine, then MAL, then along with MAL comes histidine, then thymine, along with thymine it is bio, then along with bio it is ASI, then thymine, and next you have cysteine, which is at 10 minutes. So it should be between methionine and threonine. So cysteine is over here. So cysteine goes at 11 minutes. So that is correct. Now histidine goes at, now if you plus everything, that is 15, 12, 15, 17 and 9, you will get at what time histidine is going. That is your option A. Then if you see for ASI, ASI means you have to plus 9, 1 and 5 to histidine time. So you will get at what time ASI is going. So that is according to option B, and cysteine goes at 11 minutes. That is option C. So that's it. Thank you for watching. Please do like, share and subscribe. Thank you for watching.